Hello. First, I also want to thank the organizers for giving me the opportunity to participate in this fantastic meeting. It's a wonderful venue and wonderful city and wonderful people. I'm having a great time seeing my friends in this meeting. And wonderful weather. Well, in Madrid we had the same. So I'm gonna give you a bit of an update what we are doing with Staph aureus communities in my lab. Initially, what we are interested in my lab, but with our motivation is to understand how bacteria are able to sense extracellular signals and translate that into a physiological response that is according and proportionate to the signal that they have sensed. And we are recently moved to study one pathogen, Staph aureus. It's a gram-positive pathogen that generates very important problems in hospital-associated infections because of two reasons. One is the capacity of this pathogen to develop resistance to antibiotics that are conventionally used in hospitals to treating staph infections. And the second is the ability of this pathogen to generate biofilms, to attach to surfaces that generate hard-to-treat infections. Here's the process, how Staph aureus developed resistance. Immediately after penicillin was introduced, Staph aureus developed resistance to methicillin as well as other penicillin antibiotics are used to kill Staph aureus. This is because Staph aureus generates the cell wall here at the tip, at the division septum. It's not like raw bacteria that is all over. In Staph aureus, it's only produced here at the tip of the division septum. And normally the cell wall involves the activity of these penicillin binding proteins. With these two reactions, they are responsible for the cross-linking of the cell wall. When penicillin binds these proteins, the cross-link of the cell wall cannot be produced, therefore they cannot divide, and that's how penicillin works. In this particular case of the methicillin-resistant Staph aureus, these strains, they harbor an additional penicillin binding protein. That is, penicillin cannot bind to this new penicillin binding protein. So that means that these strains with this new penicillin binding protein are able to grow, are able to cross-link the cell wall, even in the presence of these penicillin antibiotics. For those strains, for these MRSA strains, the antibiotic choose to kill them was vancomycin. It has a similar mechanism. It also inhibits cell division via inhibition of cell wall. But in this case, it doesn't target the enzyme, but it targets the products. It binds here to the diala pentapeptide, so it inhibits also the cross-link in the formation of the network of the peptidoglycan, not by inhibiting the enzyme, but by inhibiting the product. Unfortunately for these particular strains, it's relatively easy to find resistance in hospitals against vancomycin because, well, Staph aureus develop a mechanism in which some strains acquire a thicker cell wall. So vancomycin has a harder job to reach the target here, the cell division septum. So with thicker cell wall, these strains that are resistant to vancomycin, they do not show incredibly high resistance to vancomycin, but it's just high enough and good enough to resist these vancomycin treatments. The bad news is the mechanism by which Staph aureus can develop this phenotype, this little thicker cell wall, is by acquisition of mutations in many different genes, like probably 25 or 30. So it's relatively easy to find these particular VISA strains in hospital settings. And it generates a big problem because once a given strain is resistant to penicillin and acquires vancomycin intermediate resistant Staphylococcus aureus phenotype, it's really hard to eliminate these strains from the hospitals. The second thing is, as I said, it's biofilm formation. This bacterial species is really well known for its capacity to grow attached to surfaces that you can introduce in the body because of the patient has received surgery or probably in the bones. And those are extremely resistant to antibiotic treatments and generate long-term and hard-to-treat infections. So biofilm formation in Staph aureus is controlled antagonistically to secretion of toxins. Here's the quorum sensing system, the AGR system of Staph aureus, that is also an autoregulatory system, antagonistically controls the genes, it inhibits the genes that are responsible for biofilm formation, at the same time that activates the expression of a number of genes that are used in toxins, phenol-soluble modulins. Those are small peptides that allow Staph aureus to spread rapidly and also kill eukaryotic cells, so they can be considered toxins too, and a little antibiotic that's called bacteriocin of Staph aureus. This quorum sensing system that antagonistically controls these two processes is inhibited by a sigma factor, sigma B. This sigma factor responds to stress, cell envelope stress, we think. So when Staph aureus sends a stress stimuli, it inhibits the quorum sensing system. At the same time, it activates a general stress response that includes the production of pigment that is called staphyloxanthin, and it's yellow, and is the reason why Staph aureus has this yellow coloration, and it's called aureus because of the gold coloration of the bacterium. So when we were working with Staph aureus, we were trying to develop a biofilm formation assay to explore antibiotic resistance in these conditions. We came across two series of papers that give us an interesting idea. For one case, there was some paper saying biofilm formation is particularly prominent in niches in the body that contain a high concentration of magnesium, like bones or kidneys. So it seems like magnesium-rich conditions are promoting biofilm formation. On the other side, there was some other papers saying that tissues that they were unintentionally depleted of magnesium, they were developing acute infections. This one interesting case in the 80s in the United States, there was a company that commercialized a new brand of tampons that accidentally sequesters magnesium from the tissues, and that generates a lot of infections in women in the United States, and it causes a lot of deaths. So with these two considerations in mind and the antagonistic regulation of toxin secretion and biofilm formation in Staph aureus, we thought that magnesium can be a factor that affects quorum sensing and can be involved in biofilm formation in our system. So we developed a biofilm formation assay in which is basically Petri agar dishes enriched in magnesium. This is a normal TSV medium. So Staph aureus grows in this medium without any particular consistency. We have codes in the lab to name how it grows, Staph aureus in the plates. So when it grows without any particular consistency, we call it mayonnaise. So it gives you an idea about the consistency of the colony, but we don't eat it. When we grow it with magnesium, see, it develops these really strong structures. They're really strong. Here I show you a picture of one that we broke with a tip of a pipette, so you can have an idea that they are not flexible. They're really robust and compact structures. The code for this is cookies. So you can have a comparison between a cookie and a mayonnaise. So when we grow them in magnesium, we make cookies. So with these biofilms, it seems like they are very, very strong aggregates that they are generated with magnesium and with no other cation. So we were trying to explore first, what is the signal, what is the molecular mechanisms that the Staph aureus activates to develop these biofilms, and what are the consequences of cells, Staph aureus cells growing in these particular conditions. One thing that we found, it's biofilm formation in magnesium-enriched conditions is dependent of sigma B, because the sigma B mutant cannot develop biofilm formation, only in magnesium-enriched conditions, and we can't kind of recover when we deleted the AGRB mutant. Although this double mutant is pretty sick, so it doesn't grow really well, but we see biofilm formation recovery in these particular conditions. So it seems like sigma B is involved in sensing magnesium, the extracellular magnesium, and triggers biofilm formation. when we were exploring the idea, because sigma B sends a cell envelope stress, it's very well known that magnesium is incorporated in the cell wall or gram-positive bacteria, and it helps in the interaction between the cell wall teichoic acids, so it makes the cell wall more rigid. So we thought that probably that's what is causing the extracellular magnesium in the outside, and that's what is kind of sensing Staph aureus to develop this phenotype. This is an experiment of atomic force microscopy. What we do is we use the microscope has a needle, and then we poke the cell wall of one given cell, and we measure the force in which the needle is pressured back. So if it has a rigid cell wall, it will be pushed back with more intensity, with more force. Here you see in the TSV magnesium, we have a big lecture force back for the needle in compared to the TSV only wild type. This is a control strain that we know. It has weakened the cell wall. So it seems like Staph aureus cells that are growing in TSV magnesium, they have a more rigid cell wall, according to our atomic force microscopy experiments. This is an additional experiment which we treat cells with lysostaphine. Lysostaphine is an endopeptidase that degrades the cell wall of Staph aureus. So we think our theory, our hypothesis is that if it's more rigid, it will resist better the action of lysostaphine, which is seems to be the case. When we plate later, after the lysostaphine treatment, we plate later the cells. We see that we have more survivors when cells were previously grown in TSV magnesium than when cells were grown in TSV. While without lysostaphine, we will have, of course, we have more cells, but it's comparable, the number of survivors. So it seems like the effect in TSV magnesium is related to the cell wall. And also, we think with the presence of teichoic acids, when we apply tunicamycin, which is an inhibitor of teichoic acids in the cell wall, the differences are lower comparing TSV magnesium and TSV, which led us to generate two different phenotypes in Staph aureus. Ones that are locked in the overproduction of teichoic acids, so they are overproduced, or they are overproducing teichoic acids. And some strains that are lower in the down regulation of the production of teichoic acids. In this particular case, they respond with lower intensity to the presence of magnesium, the ones that they don't have that much teichoic acids in the cell wall. In contrast, the strains that they have a higher production of teichoic acids, they seem to respond more efficiently to the presence of magnesium in the medium, suggesting that magnesium seems like increase the rigidity of the cell wall of Staph aureus, and this is probably because it helps in the cross-linking of teichoic acids, so it makes the more rigid network. This is something that is known already from Bacillus subtilis and other gram-positive bacteria. So it is incredible how different some cells respond to the presence of magnesium. We don't have the full data yet, but there's a high variety in the number of strains and how do they feel magnesium. We don't know yet if it's because sigma B is higher produced in other strains than others. We don't know if it's because there are different concentration of teichoic acids in some strains than others, but the strains that they respond more, the strains that respond higher to the presence of magnesium, the higher response strains, they develop this sectoring phenotype that Kevin Foster was showing at the beginning today. Normally, those are the strains that they are more sensitive to the presence of magnesium in the medium. I'm going to talk about this particular strain, SC01. This is a community-associated MRSA. So MRSA normally is associated with hospitals because it has a tendency to infect immunocompromised people, but there are some strains that they are also able to infect healthy people in kindergartens, in gymnasiums, in schools, and those are community-associated MRSAs. This one is one of them, CCO1, and it's actually isolated from a bone infection. This is the look of the strain when we grow it in TSB. It has a mayonnaise phenotype. When we grow it in TSB magnesium, it develops this flower phenotype is really strong. It's a cookie phenotype, but it's like sectoring in some regions with different colors, obviously cause of the pigmentation. Here's the 5 days progression of the colony. You see at the second day there is a white strain that evolves, and it clearly grows really fast and takes into the periphery of the colony, because it grows really fast. And the day third, there is a third strain that is yellow, but not that yellow as the origin strain that is here in the core, and immediately breaks through the white sector and develops these flares that generates this flower-like phenotype. When we take samples from the white sector and the yellow sector, it develops these communities that are homogeneous, so it seems like those are stable. The strain that is the origin, the strain that is here in the middle, it keeps developing the flower in those conditions. So it seems like it is an unstable strain that develops mutants in these particular conditions, and those mutants are more stable in TSB magnesium. There are differences in the production of staphylococcal, of course, as you can see, but there are also differences in the formation of biofilm and hemolysis. So the white strain cannot develop biofilm, but it produces a lot of toxins, and the yellow strain, it produces biofilm as the wild type, but it cannot develop almost toxins. So it seems like there is antagonistic regulation of these two strains, just like what we were thinking about the quorum sensing system of Staph aureus. So we did some RNA-seq, and immediately we focused on the expression of the quorum sensing system in those strains. And indeed, we see that the white strain is actually expressing a lot of these, expressing very much the genes cluster that codes for the quorum sensing system of Staph aureus. We also monitor production of the molecule, of the quorum sensing molecule, using our orthogonal system that we generate in Bacillus subtilis, showing that the supernatant from the white strain is actually able to activate our orthogonal system in comparison to the others. So it seems like the white strain is a hyperproducer of the quorum sensing signals in Staph aureus. We think because initially we generate this biofilm assay, the initial purpose was to inhibit quorum sensing, to inhibit AGR, so biofilm can be developed. Probably we think this is a counter response against this idea of inhibiting quorum sensing in Staph aureus. So when we purify the quorum sensing signal and we add it to the plate, the flower is not formed. So it seems like when there is quorum sensing, when there is a lot of molecule in the medium, we don't get the flower phenotype. Then we sequence the 9 of these sectors to see where the mutation is. We find that many of these mutations are in sigma B. That's why the phenotype is mutant. The phenotype is white, because it cannot develop the staphylococcal, and that's why it has a superactivated quorum sensing system, and it produces a lot of toxins. In addition, we think this is the reason why the white strain grows that fast and immediately take over the periphery of the colony, because it produces a lot of these phenol-soluble modulins that they are surfactants, so they allow this colony to spread over. You see in a swarming assay how the white strain spread in comparison to the origin. We think that's the reason why it can grow faster than the origin, because it spreads over the agar plate at least faster in time in comparison to the origin. So here is the summary of this first part. We think in these particular TSB magnesium conditions, we see the development of one strain that is white, and it seems like it produces it's more efficient in producing toxins and phenol-soluble modulins, but it's not able to form biofilm. And because of this, the production of these phenol-soluble modulins, it seems like it can outcompete the wild-type strain and immediately take over the community. So we generate artificial assays, more like a synthetic colony, in which we mix another MRSA strain, in this case USA 300. This one doesn't make the flower phenotype in these conditions, and we generate a wild type and a sigma B mutant. We mix them in the relation that we find in the flower at day 2, and we plate them in TSB magnesium. And that's what we observed in these conditions, the TSB magnesium. When we mix the strains, it seems like the white colony is also able to take over, immediately grows really fast and take into the periphery and keeps growing. Similar to what we have seen with the flower phenotype of SC01. Something that is curious from this experiment is that we still have the flares of the third strain, which we didn't add it at the first place in the tube. We just add wild type and sigma B mutant. But we have the development of this third strain totally similar to the flower phenotype. So that led us to think that what it triggers the differentiation of these flares, what it triggers the differentiation of the third strain, is actually the presence of the white one. So we were exploring possibilities. What is the biological pressure that gives to the evolution of this third strain? We know that the white strain produces a lot of the toxins and phenol-soluble modulins. We cannot see in the literature anything associated, any pressure associated with production of these compounds. But we immediately got our attention to this molecule. It's an antibiotic. It's called BSA because it's bacteriocin of Staphylococcus aureus. And it is well known the biological pressure that antibiotics puts on bacteria to evolve mutations and to resist these antibiotics. So we immediately went to the RNA-seq data to see how is the expression of this bacteriocin in the white strain. It's around 400-fold expressed higher in comparison to the origin of the yellow strain. And this bacteriocin is normally cryptic in Staph aureus, or its production is pretty low. So in this particular strain, we have the production really high because it's normally cryptic. The mechanism of action is not really well understood, although people believe that it also targets cell wall synthesis and cell division in Staph aureus, probably by inhibiting the flipping of the lipid 2 to the other membrane, so it doesn't cross-link the cell wall. In any case, the outcome of this antibiotic will be very similar to methicillin and vancomycin, so inhibiting of cell wall and therefore inhibition of cell division of Staph aureus. We took supernatants of this antibiotic. We added to Bacillus subtilis biofilms to see that the supernatant of sigma B has, supernatant of sigma B has a super high antibiotic activity, but is inhibited when we deplete either AGR or, of course, the genes that are responsible for antibiotic synthesis. And the same happens when we test the supernatants from the 3 strains that we obtained in the flower phenotype. The origin didn't give us strong profile for killing Bacillus subtilis. However, the white strain gave us strong killing of Bacillus subtilis biofilms, in contrast to the yellow strain that didn't kill. So it seems like supernatants from the white strain, they are rich in this antibiotic that kills Bacillus subtilis, and counting with the 400-fold expression that we find in the RNA-seq, it seems like this antibiotic can be the bacteriocin of Staph aureus. I'm going to show you now 3 experiments that point to the idea that BSA, the production of BSA from the white strain, is responsible for the differentiation of the 3rd yellow strain. First, when we do the same artificial assay, but instead of mixing sigma B, we mix sigma B double mutant with the antibiotic, we are able to still see how the white strain grows faster than the origin, but we do not see flares coming out. So it seems like the absence of BSA prevents the formation of flares. Second, by adding purified BSA to the TSB medium, we think we can bypass the differentiation of the white one, because we go from the origin to the yellow strain that is here. and also by generating an origin mutant that is deficient in BSA, we also can get a similar phenotype with the development of the white one in the periphery, but no flares in the coming out of the origin. So when we think that this yellow strain responds to the presence of BSA, we start with purifying the 3 strains and testing the resistance of the 3 of them to purify BSA. In the case of the white strain, it shows a higher profile in resistance of the BSA because the gene expression cassette not only produces the protein, but also produces the immunity proteins that protects this particular strain against the production of the antibiotic. For the origin that was not able to express the cassette for the production or the immunity of this antibiotic, we see a very low expression profile, low resistant profile. It dies with lower concentrations of the antibiotic. However, this yellow third strain that comes in the flower, it resists intermediate concentrations of this antibiotic. So even though it does not express the gene cluster that provides immunity to this new antibiotic, because it shows an intermediate phenotype in the resistance of BSA, we test other antibiotics that are similar in the mechanism, like for example, vancomycin. When we use vancomycin, of course, now the white strain does not resist vancomycin because the immunity was specifically for BSA, so it goes back to the profile, very low profile, resistant profile with the origin strain. But the white strain, the yellow, it shows a similar profile, the intermediate resistant phenotype in comparison to very similar to the profile that shows with the yellow, with the vancomycin. So it seems like a rather broad mechanism that allows the yellow strain to resist or intermediate concentrations of vancomycin and intermediate concentrations of BSAs. Our hypothesis was that, okay, this is consistent with the idea that probably this yellow strain is a Visa phenotype. This has a thicker cell wall that allows them to resist in a very unspecific way, allows them to resist the activity of multiple antibiotics that attack cell wall, including BSA, of vancomycin. So in this particular case, the Visa, with a thicker cell wall, will prevent the interaction of BSA to the generation of the cell wall here at the tip of the division septum. So there were many experiments to test this. I'm going to show you just one of them, 2 of them. The first is we used this lysostaphine treatment that we know it degrades the cell wall to test the capacity of these 3 strains to resist lysostaphine. We treat them with a very mild treatment of lysostaphine, and then we plate them in TSV medium, and we count colony-forming units. This is the number of colony-forming units that we have on average in the flower. And when we treat them with lysostaphine and we plate them, this is the average of the colony-forming units. So it seems like in the treatment of lysostaphine, the treatment of these endopeptidase, the yellow strain is more resistant. It resists better the activity of these endopeptidases in comparison to the other 2 strains. That is consistent with the idea that it has a thicker cell wall or has a stronger cell wall that prevents probably the diffusion of vancomycin. Of course, we can make experiments with vancomycin labeled fluorescently to see if vancomycin can reach the division septum or the tip of the division septum, or it stays here at the septum, stays in the surface of the cell, in the cell wall of the cell wall of the cell, where there is no cell wall synthesis coming. So we were counting several hundreds of cells where is the vancomycin observed, and making the statistical analysis. So it seems like the cells in the yellow strain that accumulates the fluorescence at the tip of the division septum is significantly lower comparing to the origin or the white strain. We also try other approaches like electron microscope, but with electron microscopy you can see there is a thicker cell wall, but also depends on the edge in which you're doing the sectioning. So it's it's kind of inconclusive. Those are our two best assays to explore a visa phenotype in these strains. In this particular case, we also sequenced our strains. We found, as it is in the literature, we found a large variety of mutations, many of them already published that confers a visa phenotype in Staph aureus, but in this particular case of the visa phenotype, it's not a mutation in one particular gene, but a pool of 20 or 25 genes that all of them might lead to a thicker cell wall and a visa phenotype. In general, there are two-component systems, WOC, GRAS, and VRS mutations in there are known to confer a visa phenotype in Staph aureus, and we find mutation in those three. Then we go in vivo. We went to a mouse infection model to check if this flower phenotype also happens in tissues that are enriched in magnesium, like bones or kidneys. We took this SC01 growing in TSV, so we were very careful that the flower was not developed, and then we infect a cohort of mice. We took the cells, and when we plate them to see in which organs the percentage of colonies, the color of the colonies that we got. Normally, in hearts, spleens, and livers, we have a very low concentration of cells, and all these cells that we recover from those organs, they were just origin. We have no differentiation. We have differentiation in lungs, joints, and kidneys. In joints and kidneys, the pattern is very similar. So we have an origin, some of the white, and the differentiation of the visa strain is more pronounced, which is consistent with what we have seen in the flower at later stage points. In lungs, it's very interesting results. We are actually working on that now. We have a hypothesis. I have right now zero data to support this hypothesis, but I'm going to say anyways. So in lungs are different from infection because they have surfactants to expand the lungs, and these surfactants are known to interact with antibiotics to inhibit the activity of antibiotics. This is why infections in lungs are so difficult to treat, because when you treat them with antibiotics, the surfactant of the lungs inhibit them. What we think what happens here is that the surfactant of the lungs is inhibiting BESA. So it happens that it's a differentiation of the white one, and it's producing the BESA, but it's not exerting any biological pressure to the development of the third yellow strain, the visa phenotype, because the surfactant activity of the lungs. So normally, when we infect mice with this particular strain, the recovery of white strains from the lungs is incredible. It's really, really high. And we are really excited about this phenotype. With the other strains, the white and the yellow that comes after the differentiation, the infection potential, this is just to show that the infection potential of these new strains is also pretty high. So these new strains are also able to infect and kill mice, just like the origin strain. So this is my last slide to show you how the evolution comes to from an MRSA strain in response to the presence of magnesium. We think it's because it generates some extra rigidity in the cell wall, and that causes stress that triggers sigma B. And in some strains, only in some strains, this seems like a strong force that drives them to inhibit the expression of this sigma B by accumulating a mutation and inhabilitate the gene. Inhabilitation of this gene develops a second strain that is overproducing the quorum sensing system and overproducing the quorum sensing regulon, including toxins, and antibiotic that is BESA, that exerts another biological pressure to the rest of the strains to develop a third phenotype that is a visa-like phenotype, similar to the ones that is found in hospitals after the treatment of vancomycin. And this is my lab in Würzburg. Some of them are already moving in Spain, having a great fun. And thank you very much for your attention, and glad to take any questions.